So, hello and welcome everyone again. My name is Ellie Arnold, and I am the training specialist from the Canadian Stem Cell Network. While we meet today on a virtual platform, I would like to begin by acknowledging the Indigenous peoples of all the lands that we are on today. The work of Canada's Stem Cell Network occurs from coast to coast and occurs on the traditional, ancestral, and unceded territory of many Indigenous peoples and nations, including the Anishinaabe Algonquin Nation, where SCN is headquartered. The Canadian Stem Cell Network honors, recognizes, and respects those nations as traditional stewards of the lands and the waters where we work and meet. I'm pleased to welcome everyone to our second session of the Global Regenerative Medicine Trainee Speaker Series, hosted in partnership with the Australasian Society for Stem Cell Research, the German Stem Cell Network, the Japanese Society for Regenerative Medicine, the Belgian Society for Stem Cell Research, and the Korean Society for Stem Cell Research. We would like to thank a dedicated committee of trainees drawn from each country for helping to organize these sessions, select speakers, and facilitate the session. So you'll see some of them today introducing our speakers. Sessions of this seminar series are currently scheduled to be held every 2 months until January. So if you're interested in attending future sessions, please check out the Canadian Stem Cell Network website to see more details and register. We'll put the link in the chat as soon as I finish speaking. I also encourage everyone to check out the training section of SCN's website and also your home country's network or society webpage to learn about other training opportunities that might be available. I just have a couple of small notes before I pass it over to our committee members to facilitate the session. The session will be recorded and posted publicly on Stem Cell Network's YouTube channel. So if you'd like to rewatch the session, or for everyone who couldn't join live, that will be available. Each speaker will have up to 15 minutes to present and then a few minutes for questions, and we will encourage you to put your questions in the Q&A chat box located at the bottom of your screen. And your participation in the discussion is highly valued, so please do engage. The closed captioning is enabled for the session, and you can choose to show or hide the automated closed captioning. And with that, I'll hand it over to our first trainee committee representative, who is from the Korean Society for Stem Cell Research, to introduce our first speaker. So Sang-Yeon, please, over to you.
Thank you, Ellie. My name is Sang-Yeon Park, and I'm based in Korea. Where are you joining us from today? Please let us know in the chat. I'm delighted to welcome the first speaker on behalf of Korean Society for Stem Cell Research. The first speaker for this session is Ji-Hyun Lee. Ji-Hyun is currently a research fellow at Institute for Basic Science in Korea and will be speaking about their work on P57 KIP2 imposes the reserved stem cell states of gastric chip cells. Ji-Hyun, please go ahead and begin your presentation.
Okay. Can you see it well?
Yes.
Okay, thank you. Thank you for the kind introduction and for giving me an opportunity to present my work in this great seminar series. Today, I would like to talk about molecular switch of reserved stem cell state of gastric chip cells. Gastritis, so gastric injury, can be caused by various damages. Helicobacter pylori infection, non-steroid anti-inflammatory drug like aspirin or alcohol can be the damage source of the stomach. If your stomach is damaged, the lining of the stomach is injured, and also the stomach is inflamed. And I'm wondering how the stem cells respond to damage. To know that, we firstly need to know where the adult stem cells are in the stomach epithelium. If you see the stomach, the body of the stomach is called corpus. Corpus has a lot of numerous glandular structure like this, and each glandular structure have four different parts from the lumen, pit, isthmus, neck, and base. And due to the fact that the epithelial cells in the stomach are having high turnover rate, people thought the adult stem cell in the stomach have to be also proliferative. So researchers found previously that the proliferative cells are residing at the isthmus, and by the lineage tracing method, they found that the stem cells are residing at the isthmus region, and they are highly active proliferating. And there are second stem cell population, which is at the base of the corpus epithelium. The cells at the base are chip cells. They are secreting proenzymes to the lumen to help the digestion. But 10 years ago, researchers found that very well-known stem cell markers like Troy or Lgr5 are highly expressed at the chip cells. So people thought maybe those are another stem cell populations in the stomach epithelium. To test that, over last 10 years, people did the lineage tracing using several different chip cell markers, and they found without injury, the cells, the chip cell cannot generate other cell type. They only located at the base of the stomach gland, even after long-term, like 3 months of tracing. But after the damage, they can generate other cell type to contribute to make up the gland during the regeneration. Due to this, chip cells are now known as reserved stem cells. And in 2019, in our lab, in my previous lab, by the random lineage tracing in homeostasis, actually the seemingly one unit of the glandular structure can be governed by 2 different stem cells. One at the isthmus by active stem cells, the other at the base, which is mostly quiescent chip cells. So in the stomach, in homeostasis, you can understand there are 2 different stem cell populations, and mostly in homeostasis, the glandular structure is mostly governed by active isthmus stem cells. But I was wondering what the function of the stem cells after injury. So I focused on the base chip cell because they can be activated very rapidly after the injury. But at the time when I started this project, we still didn't know what the molecular switch of this chip cell activation. To know that, we utilized Troy EGFP knock-in mouse line, and we injured the stomach and sorted GFP positive chip cells before injury and after injury in time course, and did bulk RNA-seq. And what we found was chip cell markers are highly downregulated after the injury and comes back to normal upon recovery. The proliferative markers are highly upregulated and comes back to normal after recovery. The stem cell markers like LGR or Troy didn't change much their expression during this regeneration process. We also performed the single-cell RNA-seq after sorting the stomach epithelial cells. And what we found was the chip cell and highly proliferative isthmus stem cells are having very different characteristics. So they are mapped in a UMAP very far from each other before injury. But after injury, most of the chip cells now have their characteristics very similar to the proliferative cells. So it means chip cells change their characteristics a lot after the injury. And these injuries responsive chip cells had very unique characteristics. Not only the chip cell markers, but also base stem cell marker and even other cell type markers were all expressed intermediate level or high level in the injury responsive chip cells. meaning that this chip cell change their characteristics to more uncommitted progenitor after the injury. We could confirm this data from the single cell by immunohistochemistry. After the injury, before the injury, you can see each cell marker, chip cell markers are at the base, and next cell marker in green are at the in the upper part of the chip cell. And actually, you can see the proliferative cells at the isthmus region. But after the injury, those markers expression is are all mixed at the base region. So now at the chip cell region, they co-express several different cell markers by the injury. From these bulk RNA-seq and single-cell RNA-seq data, we found a great candidate of the molecular switch of chip cell activation, which the molecule is called p57, and p57 is one of the cell cycle inhibitor. Because after the best non-characteristic change after the injury was in the chip cell was the proliferation change. So we thought this would be the great candidate to study further. And the p57 expression is high in the chip cell without injury, but after the injury, its expression gets low. You can also see that from the bulk RNA-seq data. So its expression gets low after the injury and comes back to normal. And we confirmed that data from the immunohistochemistry. To test the functionality of the p57, we utilized organoid system. So we established gastric organoid and introduced p57 overexpressing organoid by dox-inducible manner. Because if we just see the in vitro culture system, cells are very highly proliferative. So we decided to overexpress p57 in the organoid by doxycycline treatment manner. And we checked the line has established well. So only after the doxycycline treatment, we could see p57 was expressed, and it could be visualized very well by M-Cherry. And Ki67, one of the proliferation marker, was highly downregulated. After setting up this organoid, we wanted to check whether p57 expression per se in organoid can induce the long-term quiescence in the stem cell in vitro. Because if you think of the in vivo situation, the chip cells are always are mostly quiescent in homeostasis. So we wanted to mimic this kind of homeostatic condition by treating doxycycline for long term. So before the doxycycline treatment, we could see the organoid could grow very well, so that we needed to split the organoid after every week. But once we treated doxycycline, so overexpressed p57, after some time, the organoid didn't grow much, so they don't change their growth, or they they don't grow until more than 3 months. To check whether the cells in the organoid, stem cell in the organoid, maintain their stemness more than 3 months of p57 overexpression, we split the organoids and withdraw the doxycycline. And surprisingly enough, we could see the organoid could grow again, and even we could split the organoids. This means that p57 introduction to the organoid can induce the stem cell quiescence without losing their stemness. Furthermore, I wanted to see whether p57 introduction per se in the organoid can change other aspects, like mimicking in vivo situation. So after treating doxycycline, I checked the ultrastructure of the organoids, and I could find that there are more secretory granular structures, which is similar with the chip cell function, because chip cell secretes the enzyme to the lumen. We could also confirm that the chip cell markers are highly upregulated after the doxycycline treatment, so after p57 expression in vitro. This means that p57 expression per se can induce stem cell quiescence, but also can induce chip cell maturation. After checking this functionality, we moved to in vivo situation. So we confirmed the p57 functionality in vivo. To do that, we injured the tissue, injured the stomach, and at the same time, we overexpressed p57. Without p57 expression, upon injury, you could see this co-expression of several different cell markers. But when we overexpress p57, this co-expression was highly downregulated, which means the p57 can inhibit the induction of the injury response. Due to the time limit, I cannot show you the knockout phenotype, but we could also see the prolonged injury response without p57 in the stomach epithelium in vivo. So in summary, now we in the stomach in homeostasis, chip cells are mostly quiescent, but now we know the p57 can be the switch, so it is on in homeostasis, but after the damage, p57 get turned off, then the chip cell can be activated to regenerate other cell types. Yeah, with that, I would like to thank my previous lab members at IMBA in Vienna, especially my previous supervisor, Dr. Bong-Kyung-Koo, and my other lab members, and our collaborator in the world. Now I move to my position to IBS in Korea, and now I have my own team, and we are now exploring a lot of interesting phenomena in the stomach and related to the stomach stem cell. So if you are interested in stomach stem cell field, please contact me. I will be happy to discuss. And thank you for your attention, and I'm happy to take any questions.
Thank you, Ji-Hyun, for a fantastic talk. This was a very nice overview of your work, and I'm so excited to see what comes next. We now have time for a few questions from the audience. To ask a question, please put your question in the Q&A chat box, and I will read the questions out loud to the speaker. Okay, I'm gonna first question. Is it possible, or is there any knowledge that p57 plays a similar role in different type of other stem cells in the intestine or other part of the body as well?
Yeah, thank you for the question. Due to the time limit, I didn't really focus on that part, but yes, p57 is known to induce the long-term quiescence in hematopoietic stem cells and also in neural stem cells. And very recently, when I published this paper, actually in other group in Japan, published similar paper about the role of p57 in intestine. So in quiescent stem cell in intestine, p57 expression is also high. So it was very, very interesting and surprising. So I think the functionality of p57 in adult stem cell can maybe general.
Thank you. Okay, I have one more question. Maybe it is possible for chip cells to transition into injury responsive chip cells through similar mechanism in stomach injury caused by Alcor or Helicobacter pylori. Yeah.
Yeah, so thank you for the nice question, Sang-Hyun. So we didn't really test. So the injury model we used is the model that depletes a parietal acid-secreting parietal cell in the stomach, and it is very well-known model, and it mimics actually Helicobacter pylori infection in human. But there are some models, injury models, like alcohol introduction, and actually we didn't test about it, but we are actually having another model which I cannot really tell you now what the model is, but we actually saw very similar phenotype of the chip cell activation in other models. So yeah, we are actually working on it.
Oh, okay. Thank you for answering. Yeah. Oh, okay. Thank you again, Ji-Hyun, for taking time out today to share with us a little bit about your work. We encourage interested audience members to contact Ji-Hyun to keep the conversation going. I will now hand it over to Bradley Ballaton to introduce our next speakers.
Thank you, Sang-Hyun. My name is Bradley Ballaton, and I'm happy to welcome the next speaker on behalf of the Belgian Society for Stem Cell Research. The second speaker for the session is Ben van der Veer, and Ben is currently a PhD student at KU Leuven, and he will be speaking about his work on the dual functions of TET1 in germ layer lineage bifurcation, distinguished by genomic context and dependence on 5-methylcytosine oxidation. Ben, please go ahead and begin your presentation. You're still muted, by the way.
Thank you, Bradley, for the very kind introduction, and thank you for the organizations of the speaker series for giving me the opportunity to present my work. And like Bradley said, it's about the dual functions of TET1 and germ layer lineage bifurcation, distinguished by genomic context and 5-methylcytosine oxidation, and that's an entire mouthful of words. But hopefully at the end of my talk, you will understand a little bit more about that. And basically my project, and let's see if I can get a laser pointer, which makes things easier, is about gastrulation, where we go from the single layer, single cell type from the epiblast towards the ectoderm or the primitive streak, which later will go into mesoderm and endoderm. And throughout this process is regulated by signaling such as Wnt and nodal signaling. Wnt and nodal signaling inhibit ectoderm formation and they promote primitive streak. And also at the center of this, we have our gene of interest, TET1, which is supposed to be regulating this lineage bifurcation switch. And basically the question for my project is, how is its catalytic function of TET1, because it's a DNA demethylase, so it demethylates 5-MC into 5-HMC via oxidation. How does this regulate this lineage bifurcation switch, and how does it drive cell fate decisions? And in order to answer this question, what we use is actually a very simple system where we take mouse embryonic stem cells and we differentiate them over 5 days towards neuroprogenitor cells by the addition of a little bit of BFGF, and after a couple of days we stop that. And so what we do is we take wild type and TET1 knockout cells and we differentiate them and we see what's happening. And so first what we did was bulk RNA-seq, and what we see is actually from day 0, the wild type and the knockout cells, they are still together, and from day 2 onwards they start to segregate, where and actually also we did single cell RNA-seq in order to better study this. And what we see indeed also is that at day 3, the wild type and knockout cells are still clustering in the same cluster together, which sort of also mimics, matches with this bifurcation over here. And it's interesting because at day 2, we still have all the expression of the DNA methylation machinery, but after that, this is all downregulated. But we do consider this to be sort of the prime primitive streak, because what we later see is that the wild type cells, they go towards ectoderm, and the knockout cells, they go to mesoderm and endoderm. And so we wondered, okay, how is this happening? And so what we saw is actually indeed is that in the knockout, we get an upregulation of Wnt and nodal signaling. So we thought, hey, what if we inhibit this, would this rescue it? And which one of the two is dominant over the other one? So we did the same differentiation time course. We gave the Wnt or nodal inhibitors, and what we saw is indeed that if we both give ZAF, which is a Wnt inhibitor, or SB, which is a nodal inhibitor, we saw actually that the knockout cells, that they go from the mesoderm and endoderm over here, that they all form basically ectoderm. However, there were still some differences where actually with the Wnt inhibitor, actually we got a forebrain population, while with the nodal inhibitor, actually in the knockout we do get ectoderm, sorry. But it was mainly midbrain and hindbrain cells. And so for this, we actually think that Wnt signaling in this case is inhibited by TAT1, but it's dominant over nodal signaling. So first you get Wnt signaling and then nodal signaling. But how is this Wnt pathway activated in TAT1 knockout cells? And so for that, we actually had a look at the regulons that were, or transcriptional networks, that were activated in epiblast cells and TAT1 knockout cells. And we saw a bunch of different transcription factors that we could find over there. But to our interest was one factor, TCF7L1, which is a canonical Wnt repressor. We saw actually that it was downregulated in knockout, and this is gene expression, so it's not that much, but the gene regulatory network activity is completely gone in the knockout cells. What further interests us is that actually saw that it is a TAT1 regulated gene. TAT1 is in the promoter. And so in order to prove this hypothesis, what we did is actually we overexpressed TCF7L1 in differentiation. And what we see indeed is that at day 3, but in TAT1 knockout cells, that we get upregulation of brachyury, mesendoderm, primitive streak mark, and also FOXA2. But if we overexpress TCF7L1, this gets downregulated or does not get upregulated. And also the Wnt activity gets reduced in these knockout cells, confirming that indeed TCF7L1 is one of the starters of this signaling cascade, causing this different phenotype. But this does not really answer yet the question about whether this is dependent on the catalytic activity, yes or no. And so for this, I made a catalytic mutant. So these are cells that do overexpress, or that they do express TAT1. We mutated it endogenously, but they don't have its catalytic activity shown here with this dot plot. And so in order to address this question, what we wanted to see is, okay, how do these cells then differentiate? And as you can see, our mutant cells, they actually, in contrast to the knockout cells, they form this big blob over here, which I can tell you is ectoderm cells. But we also see a higher percentage of cells, at least compared to the wild type and the mock affected cells. Also, we can see some mesoderm and endoderm cells. So actually, the mutant cells, we sort of get this tri-lineage potential where they both form ectoderm, but also mesoderm and endoderm. But how can this be? How is this happening? So it seems basically that the non-catalytic function is mainly dominant, but there's also some catalytic activity that's important for this. And so basically what we saw, and also based on the signaling, and I'm leaving some parts out of this just for brevity, is that we know that lefty is regulated by its catalytic activity, because we know that that the lefty locus is demethylated by TAT1, but TCF7L1 is regulated non-catalytically, but these both inhibitors, they inhibit Wnt signaling and nodal signaling. In a TAT1 knockout, if we then inhibit these both sides, the catalytic and the non-catalytic pillars, we get Wnt signaling and nodal signaling, inhibiting ectoderm formation and activating or inducing mesoderm and endoderm. But if we have the catalytic mutant, which does not have any catalytic activity, we do still get upregulation of TCF7L1, which kind of inhibits Wnt signaling. But because we don't have the catalytic part, we do still get nodal signaling, and that's why we get a little bit of upregulation of Wnt and nodal signaling, because there is a feedback loop. which then does not inhibit ectoderm formation, but does induce a little bit of mesoderm and endoderm formation. But molecularly, we don't really understand this yet, so we need to start looking at the different epigenetic marks everywhere. So what we did is we performed whole genome bisulfite sequencing to find differentially methylated regions that are dependent on that one. And the first question we asked is like, okay, where are these? Are these at promoters or are these at other regions, at enhancers? And so for this, you need to understand a little bit more about the anatomy of a gene, where the majority of the CpG islands, the CpG dense regions, they are in promoter or proximal, while the distal regions are less CpG dense. And these regions usually we call enhancers. Which also regulate the activity of a gene. And what we saw actually is that about 50% of all our TET1 dependent DMRs that they were and the promoters of genes, but 50% was also at enhancers. So we think that one regulates both of these. But what does this kind of have an effect on the cells? And so first what we did was ataxy, because we thought, hey, DNA methylation, it regulates the chromatin accessibility. And what we saw is actually at day 2, just before the segregation of the wild-type knockout cells, we actually don't see a difference yet. There's no, the chromatin accessibility is the same. Although there are already differentially methylated regions in the TET1 knockout cells and in the catalytic mutant cells. But at day 3, we actually see that there are some regions in the knockout that are closing down. But and in the knockouts are also opening up, but not in a catalytic mutant, despite that there are changes in DNA methylation. What we also saw is that these regions that are closing down, that these are direct TET1 targets, but the ones that are opening up are not. And as well is that actually the open and knockout regions, they are not differentially methylated yet in the mutant and the knockout, but in the regions that are closing, they are. But this suggests that there is actually a uncoupling of DNA methylation and chromatin accessibility, because again, in the mutant, they're methylated, but they're not closing down. Well, what are these regions? We think just because of the presence of the CpG density, so it's not very dense, so no CpG islands. That these are enhancers. We've also done some other ChIP-seq in order to prove this. But what was interesting is actually that these top regions, that these are mainly enriched for ectoderm motifs, but these bottom regions, these open and knockout regions, are not. And also, so we actually think that these are ectoderm enhancers and these are endoderm and mesoderm enhancers. So these ectoderm enhancers are directly regulated non-catalytically by TET1, although it doesn't demethylate, but these bottom regions not. And then I will quickly skip through some slides because I think I'm a little bit close on time, is actually where we had a look at also at polycomb regulation. And what we saw is actually that on day 0, there is an antagonism between TET1 and polycomb. But at day 5, actually, we get a gain in polycomb enrichment. There's also is inhibited, but it's not catalytically. Although if we give Wnt inhibitors again, this relationship goes away, suggesting actually that it is partially also regulated by Wnt and its catalytic activity. And on that, because I have some more data, but I will skip through it just because of brevity. I would like to conclude with this slide where sort of we mapped out its catalytic and non-catalytic activity of TET1 throughout differentiation. And as you can see, it is very complicated about when it's doing something catalytically and when it's not. And if you want to read more about it, you can check our paper over here. But on that, I would like to thank you for your attention and also acknowledge all these people, our current lab members, but also our collaborators, which helped us a lot. So that, thank you for your attention. And now I will, if there is still some time, I will be able to answer questions.
Thanks, Sven. Are there any questions from the audience? I guess if not, I'll ask a question of my own. Have you looked at, for the endoderm or mesoderm differentiation, if there's a role for like TET1 there? Like, is there from other studies that TET1 is being upregulated or downregulated between these different lineages?
Well, so we know that I think for mesomeninum, the other TET enzymes are also important. So I mean, for instance, TET2 is very important for the hematopoietic system, but that's more in adult mice. What happens during development is that especially TET1 is very highly expressed in the epiblast, 5.5, 6.5, and then they're basically all being downregulated. And then later during, let's say like 10.5, 11.5, you sort of get a slight expression of all three TET enzymes. So not specifically like that are very specific to these tissues, but more in general.
I see another question here from Pascal. It says, very nice talk and interesting mechanism. Do you know if this is a conserved signaling mechanism across species?
This is actually something that we're now figuring out. So we're actually now doing something the same in the human cell system. Of course, all the different signaling pathways, as in Wnt pathway, nodal signaling, these are very conserved between species, at least between mouse and human. With regards to TET1, of course, TET1 is also expressed in human cells. And a little bit more outside of that scope of this system, because this is very simple gastrulation, is actually that we did where we actually see that loss of TET1 in mouse cells or in mouse embryos, that it leads to neural tube defect, so that the neural tube doesn't close, which is an ectoderm defect. But recently, actually, with one of our collaborators, Richard Fennell from Baylor College of Medicine in the United States, in Texas, is actually we started screening neural tube defect cases in humans, and actually there we also found a few cases where there's actually a TET1 mutation in these neural tube defect cases, skinny bifida cases. But what is interesting is that these are co-localized or co-occurred with Wnt target mutations. So actually, that does suggest that there is some species conservation within these pathways that we're talking about. But very good question. Thank you very much.
And one last question. Would it be feasible to, this is from Cameron McKnight. Would it be feasible to generate an ectodermal induction system via overexpression, like what was done with NGN2?
Definitely. I think we should be able to do that. Although, at least for TET1, it is very tricky to overexpress it. We've also tried it a couple of times, and sort of in mouse cells, you can stably overexpress it, but in differentiation, it becomes much more tricky somehow, just because of the gene is so big. I think what would be also more interesting, at least to answer this question, is actually to make like lineage specific knockouts and also time dependent knockouts, and also actually time inducible catalytic mutants and things like this, to sort of really start to figure out what is the specific role in the different tissues. That having said, is of course TET1 is not expressed in ectoderm anymore. It is really expressed early on during like the first few days of differentiation and also first few days in development, not later anymore. So it's really an early epigenetic effect that's happening. But I definitely agree that we sort of need to go to these stage-specific knockouts. So thank you.
We're running out of time, so thanks again, Ben, for your presentation, and we encourage interested audience members to contact Ben to keep the conversation going. I'll now hand it over Pascal to introduce the next speaker.
So thank you, Bradley. My name is Pascal and I'm based in Germany, and I'm excited to welcome the third and last speaker of this session on behalf of the German Stem Cell Network. And our final speaker is Julia Wöstefeld, and she is currently a postdoc at the Institute of Human Genetics in Essen, and she will be speaking about her work on RB1 negative organoid models, RB1 negative organoids model retinoblastoma with proliferation of cone photoreceptors and loss of retinal cell type differentiation. So Julia, please go ahead and begin your presentation.
So thank you very much. Let me share my screen. I hope you can all see it. So first of all, thank you very much for the kind introduction, and also thank you for the opportunity to present our project here, which is about modeling retinoblastoma using RB1 negative organoids. So retinoblastoma is the most common eye cancer occurring in early childhood, and it's most often recognized in flash photographs of the affected children, where here the, hope you can see my mouse here, where in the healthy eyes mostly appear red, because here you can see directly through the pupil onto the blood vessels, and affected eyes appear white, because here you can see directly through the pupil onto the tumor. Retinoblastoma is caused by a biallelic inactivation of the tumor suppressor gene RB1. And there are also different forms of retinoblastoma, for example, non-heritable retinoblastoma, where the child doesn't inherit any RB1 mutations from the parents. And therefore, two mutations are needed in a specific cell of the eye to form a tumor. In heritable retinoblastoma, this is a little bit different, as the child already inherits one mutated copy of RB1 from the parents, and therefore everybody's cell carries this mutation, and therefore only one mutation or one additional mutation is needed on the other RB1 allele to form a tumor. And this mutation has to happen in a specific cell of the neuroretina, which is here the inner lining of the eye. And the neuroretina consists of 7 retinal cell types presented here. And the presumed cells of origin in retinoblastoma are cone precursor cells that develop into these cone photoreceptors that are responsible for color vision. So as the current models modeling retinoblastoma are very insufficient, especially for the analysis of early stages, we wanted to create a different model. So first of all, we have at the moment RB1 retinoblastoma cell lines that display end stages of retinoblastoma development. We have fetal retina explants that are, of course, of limited resource, and therefore research is also limited. And there are mouse models, but the problem with mouse models is that the knockout of RB1 is, first of all, embryonic lethal. And if you create a conditional knockout mouse, you also need an additional knockout of either P107 or P130 for tumor formation. And this tumor also has then a different cell of origin. So therefore, the aim of our project was to generate human embryonic stem cells carrying a mutation on RB1 using the CRISPR-Cas9 system, and differentiate these cells into neuroretina to, in the end, form a human organoid model for retinoblastoma. So first we edited H9 human embryonic stem cells. We have modifications in exon 3 and also in exon 1 of RB1, and we have cells that carry these mutations on one and on both alleles. We also generated a new neuroretina differentiation protocol that was based on the Kubahara protocol, where the human embryonic stem cells are first seeded as single cells into 96 well U-bottom plates. And this contained also different factors to drive the cells into neural ectoderm differentiation. On day 1 of differentiation, this embryoid body forms. At day 6 of differentiation, BMP4 was added and slowly reduced in the culture. And at day 12, this would drive the cells into neuroretina differentiation. At day 12, this bright outer rim forms here, which gets thickened in the long-term culture. Here you can see it more clearly. And in this outer layer here, the neuroretina is located. And our protocol goes at least until day 152 of differentiation. Using our protocol, we could achieve the differentiation of all 7 retinal cell types of the neuroretina, and they also appear in their natural order of development, starting here with ganglion cells in the inner layer of the organoids. Then amacrine and horizontal cells develop. Later, these cone photoreceptors develop here in the outer layer, and also the rod photoreceptors develop, and lastly, bipolar and Müller glia cells. We performed a comparative differentiation into neuroretina using our different cells, so our wild-type cells, but also our heterozygous and also our knockout cells. And now I want to show you the most promising results. So first of all, we saw we stained, performed a immunofluorescent staining of the proliferation marker KS67. And we saw that in the knockout cells, we have a very high number of proliferating cells. And this could also be quantified. And there we saw that the expression or the expression of KSSCR decreases in the wild type and also in the heterozygous organoids, but stays, and shows also an increase in the knockout organoids. So there's maintenance of proliferation in these organoids. And then we performed also double staining to check which cells show the proliferation. And here I want to show you the double staining of KSSCR with markers for cone photoreceptors. Here an early marker, RXR gamma, and a late marker, cone arrestin. And what we could see here, you can see it more clearly, that we have many of these double positive cells. So many cone photoreceptors that develop, that proliferate. We also quantified it, and here you can see that around 10 to 30% of all cone photoreceptors still proliferate. So again, pointing towards this persistent proliferation in cone photoreceptors, which is of course promising, as the presumed cells of origin are the cone precursor cells. We also performed RNA sequencing of the wild type, heterozygous, and knockout organoids. And first I want to show you the PCA plot, in which the heterozygous and also the wild type organoids, they separate according to their day of differentiation, shown by the different colors. And now I will add the RB1 knockout organoids, and they cluster separately, first of all, in the PCA plot. And they also show missing separation between day 96 and 152 of differentiation. So this aberrant retinal differentiation was also, or we could also see it by performing a differentially expressed gene analysis between day 35 and 152 of the wild type organoids. And what there are 4 different clusters appear, and clusters 1 and 2, they were upregulated in the wild type organoids, also in the heterozygous organoids. And there we could find genes that are responsible for the sensory perception of light and for synaptic transmission. In clusters 3 and 4, they show a downregulation at later stages of differentiation in the wild type and also in the heterozygous organoids. And in these clusters, could find genes that are responsible for cell division and mRNA processing. If we now look at these clusters here in the RB1 knockout organoids, we can see that there's not such a clear upregulation of clusters 1 and 2 and not such a clear downregulation of clusters 3 and 4, meaning there's an aberrant retinal differentiation, and also, again, with genes for cell division, a persistent proliferation again. We could also, we also screened for different marker genes for the different cell types. And what we saw is that for the wild type and the heterozygous organoids, we could see that there's a clear upregulation of marker genes for interneurons, cone rod photoreceptors, bipolar, and also the glia cells. What we couldn't see in the RB1 knockout organoids, again, pointing towards this aberrant retinal differentiation. But what we could see that there's an upregulation in the in genes involved in extracellular matrix organization in the RB1 knockout organoids here at later stages of differentiation. And what I didn't mention before is that the wild type and heterozygous organoids at day 152 of differentiation, they are, you can still see this bright outer layer where the new retina is located. And for our knockout organoids, we have a tiny problem that the outer layer disintegrates. And this might be related to the extracellular matrix organization. And this happens around day 152 of differentiation. But what was very, very promising is when we compared fetal retina and also tumor samples using a differentially expressed gene analysis. And what we could see is that the wild type organoids and also the heterozygous ones, they look more similar to fetal retina when you look at these clusters presented here. And the RB1 knockout organoids look more similar to retinoblastoma samples, especially in these clusters here. And when you create a retinoblastoma signature gene clusters, so genes that are mostly downregulated or upregulated in retinoblastoma tumors, you can see that our tumors we analyzed, they show this signature. And what was very promising is that also our organoids at day 152, they also show an increase in this retinoblastoma signature compared to the beginning stages. So to sum this now up, we inactivated RB1 in H9 human embryonic stem cells using the CRISPR-Cas9 system. We also established a modified neuroretinal differentiation protocol where all retinal cell types develop during differentiation. And we developed a human organoid model for retinoblastoma, where RB1 knockout organoids show a gain in this retinoblastoma signature. But the longer culture, as I showed you, was not possible due to the disintegration. And therefore, we need to optimize our protocol for, especially cell culture maintenance of these organoids. With this, I will come to my last slide. I want to thank a lot of people, especially Dr. Denise Kanma and Professor Dr. Laura Steenpaas for their supervision in this project. And I would also like to thank you for your attention.
Yeah, thank you, Julia, for the very interesting talk. And I can wait for to hear more in the future on this project. So now we have time for a few questions from the audience. So to ask questions, please put your questions in the chat box, and I will read the questions then aloud. But maybe I start with the first question that came to my mind. So I was wondering if maybe also other genes might be involved. So because you said in the beginning that there's this double hit in this RB1 gene. So I was wondering if there in some patients also other genes are involved, or maybe other genes are affected during this like kind of second hit.
This is something, so the problem with retinoblastoma, it's a tumor suppressor gene. So therefore, of course, you need both RB1 genes, or you need knockout of both RB1 genes for tumor formation. But there are also patients, I think, with an upregulation of MYC-N.
Interesting. There is a question by Cameron McKnight. Have you investigated the metabolism of these knockout cells? It would be interesting to see if they are primarily glycolytic like the tumor cells.
So we didn't check that. I think we didn't check that, but this, it's very interesting, of course. And maybe we could check that in the future.
Okay, thank you. And maybe a last question that also came to my mind. Based on your model and the research, do you think there you can also develop treatment options for the patients?
I mean, in the end, of course, we try to model retinoblastoma. Then in the end, of course, for us, it's also always basic science and basic research. But of course, in the end, the main goal might be that you can use this model for, not for treatment, but for, checking for different things to treat retinoblastoma with. But I think this has still a long way to go, to think about this.
Okay, perfect. Thank you so much. Thank you again, Julia, for taking the time out today to share with us a little bit about your work. And of course, we encourage interested audience members to contact Julia to keep the conversation going if there are additional questions that are coming up. So that brings us to the end of our 3 talks for this session today. So I would once again like to thank all speakers, Ji-Hyun, Ben, and Julia, for their excellent presentations today. I'd also like to thank everyone in the audience for their attention and all of the networks who are partnering on this speaker series to give a platform to the rising stars in stem cell and regenerative medicine research. And we hope you are feeling inspired because the future is very bright. Please be sure to check out the Stem Cell Network's website for our upcoming sessions of this series, which will be happening next in, I think, 2 months from now in November. And on behalf of the Stem Cell Network and the partner networks from Canada, Australia, Germany, Japan, Belgium, and Korea, thank you all for attending, and all have a great day, a good morning, a good afternoon, good evening, wherever you are today.
Thank you, everyone. And thank you so much, Pascal. That was fantastic.